about transformation. And so we've already learned about transformation in lecture. If you took Biology 150 last semester, you learned about transformation as well. But we are going to go into more detail about transformation, in particular with the experiment, and show you how that's all done. So this is the handout on transformation. And the organism that we use is E. coli. And we're going to use a special plasmid, PGLO or PGLO. Remember, we talked about how bacteria naturally absorb short linear pieces of DNA from the environment. And that process is called transformation. But in the lab, we do not work with short linear pieces. We work with circular pieces of DNA called plasmids. The problem is the bacteria do not normally absorb plasmids from the environment. And so we have to force them to absorb them. So we have to make the bacteria competent to absorb the plasmid. And the way we do that is by using heat shock and calcium chloride. And these two treatments make the membrane permeable to allow the plasmid to enter. Now, when it comes to determining if transformation has occurred, the plasmid always includes an antibiotic resistance gene. And so the idea is if the bacteria have been transformed, they now contain that gene. And so they can resist certain antibiotics. And so we grow our results in the presence of an antibiotic. If the bacteria grow, then they have been transformed because they're now resistant. And if they have not been transformed, they should be killed by the antibiotic. So we always use antibiotic resistance as the way to select for or transform bacteria. So the plasmid we're using is PGLO. It does contain 3 genes of interest: the GFP gene, which stands for green fluorescent protein, the ARAC gene, and the BLA gene. So ARAC codes for protein, and BLA codes for an enzyme, beta-lactamase. And beta-lactamase is an enzyme that destroys antibiotics in the penicillin family, which includes ampicillin. And so ampicillin is the antibiotic we're going to use to select for our transformed E. coli. So the green fluorescent protein gene actually comes from a jellyfish. So there are many sea creatures that produce these beautiful fluorescent pigments. And so they took the green one out of a jellyfish and cloned it into bacteria, so it's on the plasmid. And when it comes to the regulation of gene expression, that's one of the things we're going to explore today. So not only will we observe simple transformation and antibiotic resistance, we're also going to explore the regulation of gene expression. Because as we're going to see, not all of the transformed bacteria will produce the green fluorescent protein. Even though they have that gene is going to be turned off. And the reason why that gene is turned off is because the green fluorescent protein gene is under the control of a special promoter, PBAD or PBAD. And so PBAD, along with the ARAC gene, will actually come from the arabinose operon. So we talked about operons. And when it comes to the sugar arabinose, since you probably have never heard of that sugar, it's not a very common one. So the arabinose operon is normally going to be turned off. Only if very specific conditions are met will the arabinose operon get turned on. And it's actually a very complicated operon. But only 2 pieces from that operon are being used here. And those 2 pieces are the ARAC gene and the PBAD promoter. And so PBAD, that promoter, is normally turned off. Because remember that RNA polymerase hops onto promoters so it can begin transcription, right? Every gene has a promoter at the start, and that's where RNA polymerase hops on so it can begin transcribing that gene. Well, ordinary promoters are on, and so the RNA polymerase can hop on and start transcribing, but PBAD is off. RNA polymerase cannot hop on and start transcribing. So the green fluorescent protein gene has that special PBAD promoter. So that gene is turned off because the promoter's turned off. And we're going to learn later how to turn on that promoter so the GFP gene can be transcribed and translated, right, to make the green fluorescent protein. So here is our plasmid. So remember, the three genes we're concerned about are the ARC gene, the green fluorescent protein gene, and the beta-lactamase gene, which is called AMPR for ampicillin resistance gene, but it's a gene that codes for beta-lactamase. The beta-lactamase gene and the ARAC gene are under the control of ordinary promoters. So RNA polymerase can hop on and start transcribing. But the GFP gene has a special promoter, PBAD, or in this case, it's the arabinose BAD, promoter, or just PBAD for short. So we're going to stick this plasmid into the bacteria by heat shocking them while they're in calcium chloride solution. That will make the membrane permeable so the plasmid can get in. So any questions so far?
So the GFP is the only one without a promoter?
Nope, all genes have promoters, but those promoters are ordinary promoters where the RNA polymerase can hop on and immediately start transcribing. But the PBAD is a special promoter. It's actually going to require two things to turn it on. And so that promoter will be in the off position until it gets turned on. And so that means the GFP gene is off, not being transcribed.
Okay, thanks.
So for the procedure, what you would have done in class is each group would get two microfuge tubes, those little tiny tubes. You label the top of one plus and the other minus, and that's just to indicate that one tube will eventually get plasmid DNA and the other tube will not get any DNA. And then you use a micropipetter to add your calcium chloride transformation buffer to each tube, and then you put the tubes on ice. Then we use these sterile disposable plastic loops to obtain some E. coli. And you need two loops, one for each tube, right? Because the tubes are not sterile and the calcium chloride is not sterile. So after you get some E. coli, you add it to the plus tube and then dispose of that loop and get another one and add some more E. coli to the minus tube. And then we may vortex the tubes just to mix up the E. coli, give a nice even suspension. But we keep them on ice. And then when you're ready, you come to me and I would add 5 microliters of the plasmid to the plus tube only. Remember, plus means there's DNA in it. So the minus tube has no DNA. And then we keep both tubes on ice for 10 minutes, so nice and cold. After 10 minutes, you are going to, well, while you're waiting that 10 minutes, you get your 4 types of agar plates that we're using. And the nutrient medium that we're using is called LB. It stands for Luria Bertani, the 2 scientists who came up with this. And it's just the standard type of agar that's used for transformation. So yeah, 1 plate, it's just the plain LB agar, that's it. The 2 plates are the LB agar plus ampicillin. And then the last plate is LB agar plus ampicillin plus arabinose. So again, you have 4 plates. So one is just plain LB agar, nothing else in it. 2 of the plates are LB agar with ampicillin. And the last plate is LB agar plus ampicillin plus arabinose. And you label just the plain LB agar plate and one of the ampicillin plates minus, and then the other LB ampicillin plate and the ampicillin and arabinose plate, you label plus. So 2 plates will be for the minus tube and 2 plates are for the plus tube. So after your 10 minutes on ice, you're going to heat shock the bacteria by floating the tubes in a 42 degrees Celsius water bath for exactly 50 seconds. And so this is something that you would have had to coordinate because the timing is critical. If the amount of time in the heat shock is too much or too little, it will affect the efficiency of transformation. So you will not get good transformation if you're not long enough or you're too long. After the 50-second heat shock, you put the tubes back on ice for 2 minutes, right, to cool down, and then you put them on the desktop in a rack at room temperature. Then you're going to add some nutrient broth to each of those tubes. So we have some LB broth, it's the nutrient broth. Add that to the 2 tubes, wait 10 more minutes. Then you can put samples onto the appropriate agar plates, and we spread them with these plastic spreaders. And students always love that because you dip the plastic spreader into ethyl alcohol, and then you just ignite the alcohol in the flame. You don't hold the plastic spreader in a flame because it's plastic. It'll melt. You just dip it in the alcohol, ignite the alcohol in the flame, and then let the alcohol burn off. And now your spreader has been sterilized. And then you would spread your solution all over the surface of the agar plate. You would add the contents of the plus tube to the plus plates and from the minus tube to the minus plates. And then we incubate them for 24 to 48 hours and examine the results. So that is the procedure that we would have followed to heat shock the bacteria along with calcium chloride to make them permeable so the plasma can get in. And then we cool them down, leave them at room temperature for 10 minutes after we added some nutrients to the tube. So we gave them food and time, and then we spread samples on the surface of the agar plates. And this is what we expect to see. So remember, we have our LB plates, LB ampicillin, another LB ampicillin, and the LB ampicillin arabinose plate. These 2 plates are just plain E. coli, no plasmid. These 2 plates have the plasmid. So we see lots of growth all over the surface of the LB plate that's just plain E. coli, but we have nothing on the plate with ampicillin. So what does that mean on this plate if nothing is growing? So what does this plate mean? So somebody tell me.
Was there not any transformation?
Remember, these are the plates which have just E. coli on them. No DNA was added to the E. coli, right? So it's just plain E. coli. So there's lots of E. coli here, none here. What happened?
The operon didn't turn on?
There's no operon here. It's just plain E. coli.
Oh.
What's in the agar?
Because there's no antibiotic.
Oh no, there's lots of antibiotic. Right? The amp means that there's ampicillin in this plate, and there's no growth. So what does that mean?
Oh, did the antibiotic kill the bacteria?
You're right. The antibiotic killed the bacteria. So what do we call these two plates in this experiment? What do we call these plates in this experiment? I know for sure you guys learned this in 150.
Are they the positive?
Well, they're controls, right? They're controls. So this plate is especially important because it tells us that E. coli is not resistant to ampicillin. That's important to know because remember, how do we know that transformation occurred? We know because the bacteria have become resistant to the ampicillin, right? That's how we select for the transformed bacteria. So we have to prove that the bacteria are not already resistant. Otherwise, our experimental plates don't mean anything, right? Because we know the transformation occurred because we're giving the bacteria antibiotic resistance. If they already have antibiotic resistance, then we do not know if transformation has occurred. So does that make sense?
Yes.
So then here we have the experimental plates. So remember, plasmid was added to these E. coli. Again, both agar plates contain ampicillin. Now we see growth in the presence of ampicillin. So we know that transformation has occurred, right? Because these bacteria are now resistant and they're growing in the presence of ampicillin. So that's how we know that transformation has occurred. Now, this is under ordinary light, just the lights in the ceiling. And you can kind of see a slight difference in the color of these colonies. They're kind of more yellowish looking. But when we shine UV light on all these plates, look at what happens. We have all these colonies that are glowing, right? So they're producing the green fluorescent protein. So they've been transformed. We know that already because they're growing in the presence of ampicillin, but they're also fluorescent green. So that's pretty cool looking. But the question is, these bacteria were also transformed. They also contain the green fluorescent protein. Why are the colonies not green? All these bacteria have the same plasmid in them. But these bacteria are not producing the green fluorescent protein. Why not?
Is it because they don't have the arabinose?
Right, they don't have the arabinose. So remember, the PBAD promoter is turned off. It actually needs two things to get turned on. It needs the ARAC protein, which all the transformed bacteria are making that protein, so they have that protein, but they also need arabinose. And so arabinose binds to the ARAC protein and changes its shape, which allows the ARAC protein to bind to the PBAD promoter. That turns on the promoter. Now RNA polymerase can hop on and start transcribing the green fluorescent protein gene, and those cells will translate that to make the green fluorescent protein. But two things are required to turn on PBAD. The ARAC protein, which the bacteria are making, but the arabinose sugar also needs to be present. Only one agar plate contains the arabinose sugar, and that's this plate. This plate, the same plate here, you can see the LB-AMP ARA, so the arabinose sugar, is in this plate. And so these bacteria have both things that they need to turn on PBAD and start transcribing the green fluorescent protein. This plate only has one. It has the ARAC protein. That's not good enough. It needs the arabinose sugar. If we were to add some arabinose solution onto the surface of this plate, then the bacteria would start to make the green fluorescent protein. Does everybody understand the difference between these two plates and why these bacteria are fluorescent green and these are not?
Yeah. Yes.
All right, so two things are required to turn on the PBAD promoter, the ARAC protein and the arabinose sugar. Where does the ARAC protein come from?
Isn't it from the green fluorescent?
No, the ARAC protein, where does that come from? So where does the ARAC protein come from? It comes from the E. coli, right? Because they have the plasmid inside them, and the plasmid has the ARAC gene on it, so the E. coli are making the ARAC protein. Where does the arabinose sugar come from? Where does the arabinose sugar come from in this experiment?
From the environment? Like it's on the plate already?
Right. It comes from us. It's in the agar plate, right? So we have to supply the arabinose sugar to the bacteria. So your results indicate the presence of growth, right? So confluent growth here, no growth, some good growth here, colonies on both of these plates. Under UV light, only this plate here, the LB ampicillin arabinose plate, will be fluorescent green. Number of colonies, don't worry about it. That's only if we were going to calculate transformation efficiency, we would need to know the number of colonies, but we're not going to worry about that. So just whether growth is present and the appearance under UV light. So question 1 is asking about the genotype of E. coli before transformation and after transformation. So does ordinary plain E. coli have a green fluorescent protein gene?
No.
No, so we would say GFP negative. Is plain E. coli resistant to ampicillin?
No.
No, so we would say BLA negative, right? So E. coli before transformation is GFP negative and BLA negative. What about after transformation? After transformation, does E. coli now have GFP?
Yes.
Yes, so it's GFP positive. What about the beta lactamase gene? E. coli now resistant to ampicillin?
Yeah.
Yes, so it's BLA positive. So what was the purpose of transferring the plus and minus DNA tubes from ice to hot water to ice? So what did we call that? Ice to hot water to ice. What do we call that process?
The heat shocking?
Right, the heat shock. So heat shock, along with calcium chloride, made the membrane permeable so the plasmid could get inside. So that's how we made the cells competent so they could be transformed. Heat shock and calcium chloride, that's how we make the E. coli competent so the plasmid can get in and the cells can be transformed. Everybody got that?
Please, can you explain number 2 again, please?
So the purpose of transferring the plus and minus DNA tubes from ice to hot water to ice was to heat shock them along with calcium chloride that made the cells competent. And that means the membrane was permeable so the plasmid could get inside. So you got that, Christiana?
Yes, thank you.
So number 3, after we heat shocked them and then we cooled the tubes down, we let them set at room temperature. We added some nutrient broth and then waited 10 minutes before we spread samples onto the agar plates. And the question is, why did we do it that way? If we know that transformation occurs after the heat shock, why not just immediately spread the samples on the agar plates? But instead, we gave them some food and time before we spread them on the agar plates. So why did we do it that way?
Is it like a lag phase?
It is a lag phase, and can you tell me what's happening?
So I guess by leaving them at room temperature and giving them food before transformation, they're able to become, like, get ready, be stronger to do the transformation. Or more
The transformation has already occurred, right? We heat shocked and
transformation.
cooled them down, so transformation's over. Now we give them food and time. And so why did we do that? And it does have to do with the lag phase, but what exactly are they doing during that 10 minutes? So they are doing something. They're not growing, they're not dividing, not in 10 minutes, but they are doing something very important. And we need to give them time to do it. and nutrients, because remember, they're just in calcium chloride. There's no other nutrients there. So we give them some nutrient broth so they can begin to do something. What are they doing? Anyone have any ideas? So what's in the agar plates? That's important. What's in the agar plates?
The antibiotics.
Right, so ampicillin's in the plate. So what are we giving the bacteria time to do?
Time to resist it before they resist it. I don't know what they would be doing before that.
Well, that's the right idea. What they're actually doing is they're transcribing and translating the plasmid, in particular, the beta-lactamase gene. So they're making the enzyme beta-lactamase so that when we spread them onto the surface of agar plates containing ampicillin, they will survive. So we have to give the bacteria food and time to transcribe and translate, and in particular to make beta-lactamase. If we simply spread the samples immediately after the heat shock, they haven't made any beta-lactamase. So we put them on the agar plates containing ampicillin, all the bacteria will die. So we have to give them food and time to transcribe and translate and make the beta-lactamase enzyme, so that when we spread them on the agar plates with ampicillin, the E. coli will survive and grow. So does that make sense?
Yes.
Professor, please stand to make better words, please.
So remember, if we put the bacteria on the agar plates with ampicillin right after the heat shock, all the bacteria will die because they have not made any beta-lactamase enzyme. So they cannot resist the ampicillin, so all the cells will die. So we have to give them food and time to begin to transcribe and translate the plasmid, in particular the beta-lactamase gene, so they can make the enzyme beta-lactamase. And when we put them on the agar plates with ampicillin, the E. coli now survive because they can destroy the ampicillin.
Thank you.
So is that what happens in humans? They have like a lag time to to get the beta so that they can resist antibiotics.
Yeah, they have different ways of doing that, and beta-lactamase is one way that they can do that. But normally they will be producing that once they get inside of you. And by the time you realize you have the infection, the bacteria have already multiplied, and then you try and treat them with antibiotics. The antibiotics don't work, and hopefully you can find a different antibiotic that does work. We will talk about virulence and how many bacteria regulate virulence later on in the course when we get into the immune system. So question 4, what information is provided by the LB minus DNA plates? So these plates here. So the LB minus DNA plates, this is one of our controls. It tells us two simple things. One E. coli is alive, right? Because we need to have live bacteria to do the experiment. So the E. coli is alive, and E. coli grows on LB agar. So that's what this plate tells us. It's a control that tells us E. coli is alive and grows on LB agar. Right, so that's question 4. And question 5 is asking us about the other minus DNA plates. So this one here. Again, it is a control, and it tells us one very important thing. And what's that?
That the E. coli that the AMP kills the E. coli.
Right, so ampicillin kills E. coli. Or in other words, the E. coli are susceptible. In other words, E. coli are not already resistant to ampicillin. So we need to know that. Our E. coli cannot already be resistant to ampicillin, because resistance to ampicillin is how we know transformation occurred. We're giving the bacteria resistance. If they're already resistant, then we're not doing anything, right? So we have to prove that E. coli are not already resistant to ampicillin. And so there's no growth here, so that's what that plate tells us. No growth, no resistance. Well then, which plates demonstrate transformation? Which plates demonstrate transformation?
Is it the fourth plate?
Well, the plus DNA plates, right? So both of these plates, right, they both demonstrate transformation. Why do they demonstrate transformation? How do you know transformation has occurred?
Because of the presence of colonies.
Right, colonies that are growing in the presence of ampicillin, right? So these bacteria are demonstrating ampicillin resistance. And that is how we know transformation has occurred on both of these plates, because the E. coli are growing in the presence of ampicillin. So that's how we know, right? Transformation occurred. So question 7, why does the LB ampicillin arabinose plate fluoresce when the LB ampicillin plate does not? So why is it that the bacteria on this plate are fluorescent green and the bacteria on this plate are not?
Is it because it's the plate that contains the antibiotic?
No, these bacteria are producing green fluorescent protein. These bacteria are not, but genetically they're identical. So why is it that these bacteria are green and these bacteria are not?
Is it because there's arabinose?
Right, so can you explain in detail what's going
Like,
on?
doesn't the arabinose allow the GFP to be expressed?
Not by itself. What else is needed?
Is it because it contains the plasmid?
Well, it's all about the plasmid, but give me some details. How come these bacteria are green and these are not? And we got part of the answer, arabinose, but we need more details.
Does it contain the RXC protein?
Well, they both do. So somebody put it all together.
So the arabinose and the RC protein, do they bind together to produce the P bad promoter to make it grow?
Okay, so remember the green fluorescent protein gene is under the control of the PBAD promoter. And the PBAD promoter is normally turned off. It requires two things to get turned on. One of those things is the ARAC protein, which the bacteria are making because they have the gene on the plasmid. And the other thing is the arabinose sugar, which is only in this agar plate. The arabinose sugar binds to the ARAC protein. That allows ARAC to bind to the promoter. Now RNA polymerase can bind and begin to transcribe the green fluorescent protein gene so the cells can make the green fluorescent protein. This plate has both things, the ARAC protein and the arabinose sugar. This plate only has the ARAC protein. There's no sugar in the agar. So these bacteria on this plate cannot turn on PBAD. So the green fluorescent protein gene will not be transcribed. So does that make sense?
Yeah, are you able to repeat it one more time, please?
I don't know. So
Confusing.
confusing. It says so right here. It's all right here. So the PBAP promoter regulates the expression of the green fluorescent protein. The ARAC gene and the beta-lactamase gene are under the control of ordinary promoters. That means that those genes will be transcribed and translated to make their respective proteins. The green fluorescent protein gene is controlled by the PBAD promoter, and PBAD is normally turned off. In order to turn it on, two things are required, the ARAC protein and arabinose. The arabinose binds to the ARAC protein, and together they bind to the PBAD promoter. Now RNA polymerase is able to bind to the promoter and begin transcription of GFP. So the ARAC protein comes from the bacteria because it's on the plasmid, which is inside the bacteria now. The arabinose sugar has to be provided to the bacteria, and it's only in one of the agar plates, in this plate here. So
Okay.
do we understand it now?
Yes, thank you.
I know. It is a lot to put all together and keep straight. We are going to watch some videos, and one of them kind of goes through that whole process.
Question, Professor. So under the UV light, it's only the positive DNA plate that glows, right? That glows green.
Right, because the protein is fluorescent, so you have to shine UV light on it, and that makes the protein glow. On this plate here, under ordinary light, you can see kind of a yellowish greenish color under ordinary light, but you need the UV light to make it glow. Any other questions?
Professor, but
Uh-huh.
the growth in both plates with positive DNA, right?
Yep, the growth in both plates.
Thank you.
So I've enlarged this so we can see something interesting happening. So if we look in this plate here, you notice that there are a bunch of smaller colonies growing around the larger colonies. Everybody see that? These smaller colonies growing around the larger colonies?
Yes.
So we call those satellite colonies because it looks like they're orbiting around the larger colony. So here's the thing about the satellite colonies. They're plain E. coli. So these smaller satellite colonies are not transformed. There is no plasmid inside of them. But they're growing on a plate with ampicillin. We just saw that doesn't happen, right? Plain E. coli is killed. We proved that. And yet, here we have little satellite colonies of plain E. coli. How can that be? How can they be growing on plates with ampicillin when we just demonstrated that ampicillin kills plain E. coli?
Is it because the bacterial cell walls were not able to form?
I don't quite understand that.
I mean, the bacterial cell wall dies on the third plate by breaking out or something.
Well, the ampicillin kills them, but here we have plain E. coli growing on a plate that has ampicillin in it. So how can plain E. coli grow on this plate with ampicillin, but not on the other plate with ampicillin?
Are they attached somehow
Nope,
to the main one?
nope, not attached. But do you notice any satellites out here in the agar?
No.
No, they're only growing around the larger colonies. So does that help to explain what might be happening?
Say it again, please.
There's no satellite colonies growing out in the agar away from our transformed E. coli. They're only growing right around the transformed E. coli. So does that help to suggest a possible explanation? No ideas?
No, I can't think of anything.
No. So again, the clue is that the satellites are only growing next to a transformed colony. So the transformed bacteria are making so much beta-lactamase that the enzyme is destroying all the ampicillin in the agar immediately surrounding the transformed bacteria. And that creates a safe zone where non-transformed E. coli can survive and grow. So the transformed colonies are making so much beta-lactamase, the beta-lactamase destroys the ampicillin in the agar immediately surrounding those transformed colonies. And that allows for non-transformed E. coli to survive and grow. Not thrive, right? The colonies are much smaller, right, for the non-transformed E. coli compared to the transformed E. coli. But they're able to survive because they're right next to transformed E. coli, which are destroying the ampicillin in the agar. And so that allows non-transformed E. coli to survive and grow. So does that make sense?
So is there no agar in that area, or there's just there's no antibiotics?
Right, there's no antibiotic. The agar is still there. The antibiotic is being destroyed by the beta-lactamase enzyme, which the transformed E. coli are making.
That's cool.
Yeah, always happens, or almost always happens, especially if you give the plates a long enough time to incubate and grow. And that way the transformed colonies destroy all the ampicillin, and those few cells that were not transformed were hanging on. Now they're able to start growing. But they're growing only because they're next to transformed E. coli. So does everybody understand?
Yes.
Well, let's watch some videos of the process. So the first video, I will go through the process of transformation, and then we'll see some actual experimental procedures that are done the way that we would have done it or similar to how we would have done it. So let me just, okay.
Welcome. This little video is going to serve as an introduction to the mechanics of the experiment we're going to be doing this week on bacterial transformation. We're going to be using a plasmid, which we'll learn about in a moment, which carries the gene for green fluorescent protein, which was actually isolated from a jellyfish, that you can see here on the screen. Before we go into the mechanics of the experiment, we need to review a quick central framework or dogma of molecular biology. You know, everything stems from DNA, which we know DNA ultimately leads to RNA, messenger RNA, when it gets transcribed, and then when translated, results in a protein or proteins, which ultimately will lead to the expression of what we refer to as a trait. So keeping that in mind, we'll be able to keep track of what's going on during this process of bacterial transformation. First things first, we need to get ourselves sort of situated. This is a typical sketch of a bacterial cell areas, just to make sure we know what we're talking about. You have the cell wall on the outside, which will be important when we start talking about how the antibiotic resistance works and the antibiotic susceptibility. The pink squiggly line there represents the bacterial chromosome, which has all the genes that are necessary for the cell to survive. We're going to use plasmids, which are little circular pieces of DNA. They're known as extrachromosomal DNA. In this case, we're using a plasmid that's known as P-Glow. It's proprietary from Biorad. It's called P-Glow because it has the gene for green fluorescent protein, which will make the bacteria glow if they're expressing the gene that's on that plasmid. The process of transformation actually involves getting the plasmid into the cell. So taking this plasmid here and getting it into the cell somehow. We're going to be doing what's known as artificial transformation. We're going to be forcing these plasmids into the cells. This does happen naturally. Cells exchange plasmids all the time. That's how bacterial resistant antibiotic resistance can spread between cells. Once the plasmid is in there, if it gets expressed, it will produce the proteins that are coded for on that plasmid. And if it has another region that we'll see in a second, it will be able to be passed on to any future cells. This one's showing that we could produce two proteins, and this is of interest in GFP, the green fluorescent protein. We're also going to need to have some kind of antibiotic marker or resistance that we can test for so that we can select for cells that have been transformed. And we're going to use what's the gene for beta-lactamase, or the BLA gene, which will confer ampicillin resistance to the bacteria if they are indeed transformed. So that's a quickie overview of what transformation is. We'll be doing that in the lab. We need to go a little bit about how the plasmid works and all that in order for you to have a sense of what's going on this week. First of all, the plasmid itself. Here's a diagram of what the plasmid would look like. It has a number of areas that are of importance and interest to us. The first one is known as the ORI, or the origin of replication. If a plasmid is going to be of any use to a cell going forward, it needs to be able to replicate, just like when the chromosome replicates. So there is this region here, the ORI site, which is the origin of replication, where DNA polymerase would bind and make a copy of it before the cell divides. Another area of interest on here is the BLA gene, or the beta-lactamase gene. That gene codes for enzyme ampicillinase, which would give the bacteria the ability to break down ampicillin and therefore be resistant to ampicillin. Two other genes of interest on here are the ARA, or the arabinose C gene, which codes for a protein involved in the operon, which we'll see in a second, and then also the GFP gene, which is the gene for green fluorescent protein. Ultimately, we're going to be trying to get the bacteria that were not resistant to ampicillin to become resistant and be able to express this GFP gene. The way it works in something called an operon. Operon is a collection of genes and any of the regulatory sequences involved. So this entire area here that's circled is known as an operon. This happens to be the arabinose operon. It's involved in the metabolism of arabinose. The areas of interest here, there are three genes sort of downstream. If that thick dark line represents the DNA, those three areas code for three different genes involved in arabinose metabolism. They're shorthand arabinose B, A, and D for the three different gene products. There's also a region upstream of those coding genes, which is shown here underneath this arabinose C protein. That's the promoter sequence, which is where, if the gene is going to be expressed, RNA polymerase needs to be able to bind. The way it's being shown here, the arabinose C protein is bound on the promoter sequence and prevents the RNA polymerase from actually binding. So this gene would be turned off right now. An operon allows us to turn on and turn off genes. In this case, the molecule that will induce or turn the gene on is arabinose. Obviously, if arabinose is present, we'd like to be able to metabolize it, or the cell would like to be able to metabolize it. The way that works is the arabinose actually binds with the arabinose C protein and changes its shape. You see here it's in a different conformation than it was above. By changing its shape, it actually exposes the promoter region, which is where the RNA polymerase, which is the protein that is responsible for making messenger RNA, can now bind to the promoter sequence, since the arabinose C with the arabinose bound to it has changed its shape. The polymerase can bind and then go down the gene and actually transcribe the genes in this direction, left to right, and make the messenger RNA for the arabinose B, A, and D. It's the basic operation of our mechanics for how an operon works. In our case here, the people at Biorad have engineered the plasmid to make it a little bit different. You see on the next page here. here's what happens is they've taken the B, A, and D genes and removed them and replaced them with downstream. Here's the same arabinose C promoter area being bound to the arabinose C, but downstream of it we no longer have the three arabinose metabolizing proteins. Instead we have the gene for green fluorescent protein. So what happens is, because this operon is still controlled by that arabinose C area, when we grow the cells in the presence of arabinose, right, same molecule that will sort of turn on the gene, it will bind to the arabinose C protein and change its shape so that the polymerase can now bind. And instead of going downstream and making the three proteins involved in arabinose metabolism, instead the cell will be able to transcribe the gene for green fluorescent protein, which will then hopefully be expressed, translated into the actual GFP itself, which when we shine ultraviolet light on the bugs, they will glow green, just like that jellyfish back on the first slide. So that's the quick and dirty for how bacterial transformation works. Again, we're going to try to get this plasmid into the cells and then find the ones that are expressing both the antibiotic resistance, because they have the BLA gene, and ones that are hopefully expressing the green fluorescent protein gene. So that's what we're going to be doing this week in the lab. Thanks.
Any questions? So we have another video of the actual procedure.
In this video, you will learn how to perform a bacterial transformation. E. coli HB101 bacteria will be transformed with P. glow plasmid using a calcium chloride transformation. To start this activity, you will need an LB agar starter plate, streaked for single colonies of E. coli HB101, and 2 microcentrifuge tubes, each with 250 microliters of 0.05 molar calcium chloride or transformation solution. One of the tubes should be labeled plus and the other minus. Both tubes should be kept on ice. Flame an inoculation loop and allow it to cool. You can lightly touch the loop on an empty spot on the starter plate to make certain the loop is cool. Then gently scrape 1 large or 2 small colonies from the surface of the starter plate so that they can collect on the loop. Transfer the loop with bacteria to the plus tube and swirl it in the transformation solution to disperse the bacteria. Close the tube and place it back on ice. Flame the loop and repeat the process to add bacteria to the minus tube. Using a micropipette set to 10 microliters, obtain 10 microliters of P. glow plasmid. The volume of plasmid you will be working with is usually very small, so make certain you are looking directly into the sample as you pipette. You should also visually inspect your pipette tip to verify you have obtained 10 microliters of sample. Pipette the 10 microliters of plasmid into the plus tube. Do not add any plasmid into the minus tube. Incubate both tubes on ice for 10 minutes, making sure the tubes are in full contact with the ice. During the 10-minute wait, using a waterproof marker, label your LB agar plates according to the instructions provided with your lab protocol. After your tubes have been on ice for 10 minutes, transfer both the plus and the minus tube directly from the ice into a 42 degrees Celsius water bath for exactly 50 seconds. Then immediately place them back on ice. Incubation at 42 degrees Celsius is used to heat shock the bacteria. Incubate the tubes for 2 minutes on ice. After 2 minutes, remove the tubes from ice. Using either an adjustable or disposable pipette, transfer 250 microliters of LB broth into the plus tube using aseptic technique. Repeat this process to add LB broth to the minus tube, and then incubate both tubes for 10 minutes at room temperature. Using an adjustable or disposable pipette, transfer 100 microliters of each transformation mixture onto the appropriately labeled LB agar plate using a sterile pipette or pipette tip each time. Flame the inoculation loop and let it cool down. Spread the bacteria over the entire surface of the plate in all directions. Rotating the plate a quarter turn between spreads will ensure the bacteria is spread evenly across the plate. When finished, flame the loop and repeat this process for each plate. Turn the plates with the bottom or gel side facing up and stack the plates. Place the plates in a 37 degrees Celsius incubator for 16 to 24 hours.
And they're using those cute little mini agar plates. We use the full-size agar plates, so it really helps to use a spreader rather than a loop to spread everything around. And in the last video, we'll see some actual results. There's no sound on this video. So the BLA gene, which allows bacteria to survive in an environment of ampicillin, the ARAC gene, which in the presence of the sugar arabinose produces a protein that will turn on GFP. And the GFP, when turned on, will produce a green fluorescent protein. So there's our plasmid, ARAC gene, GFP, and BLA gene. So we're going to stick the plasmid into E. coli. So we have our calcium chloride transformation solution. We have our tubes labeled plus and minus. Remember, the plus tube gets the plasmid, minus tube does not. Then you add E. coli to each tube. Then you add plasmid, and they're just showing you when you shine UV light on the plasmid solution, there is no fluorescent green because the DNA doesn't fluoresce, right? So a green fluorescent protein gene is not actually green. The protein made from it will fluoresce. Here they're adding the plasmid, but they're just using a loop to do it instead of a micropipetter. Only in the plus tube. And then they chill it and then heat shock it at 42 degrees for 50 seconds. And then they'll cool it. Add some nutrient broth and let the tube sit for 10 minutes. Remember, while the tubes are sitting for 10 minutes with nutrients, they're transcribing and translating the plasmid, especially the beta-lactamase gene. So when we spread the bacteria on the surface of the agar plates with ampicillin, the E. coli will survive. So again, the plate that has the arabinose sugar has the fluorescent green protein because the RAC protein and the arabinose bind together and then bind to PBAD to turn it on. And that allows the RNA polymerase to transcribe the green fluorescent protein gene. So questions?
We're having a lab test next week, right?
Yep, and we'll talk about that later.
Okay, just saying.
We're also having lecture exam 2 next week. Aren't we having it next week or the week after? I forget.
I think the week after.
Yeah, maybe the week after, because you have your unknown on Monday and then the lab exam on Tuesday. I think that's what we're doing, aren't we? Yeah, so yeah, check the syllabus and see, or check the Blackboard site, and it shows what's there. I can't remember off the top of my head. Let's see. I'll just double
Are you
check.
asking about the exam, Professor?
So let me see. So week 7. Is right, the unknown on Monday, and then the lab exam on Wednesday. And so we'll have some review on Monday for the lab exam. Right, because you're not actually doing the unknown in class. So we'll have some time to review. But my old reviews, just the audio reviews, are on my web page, especially a separate review just for transformation. So anything else? You're welcome, Sarah. All right, so I'll see you guys on Wednesday.
Thank you. Bye.
Bye, Rachel. Bye,
Thank
Christiana.
you, Rachel. Bye.
Mm.
Bye, Clarice.